Hi, I'm Professor Cornelis Melief from Leiden University Medical Center in the Netherlands, and I'm one of the organizers of the 27th SITC annual meeting. I would now like to highlight a few of the sessions and say a few words about them. In the first place, we have a very exciting session on T-cell modulation strategies. As you know, multiple T cells may be involved in the response to cancer, CD8 T cells, CD4 T cells, and it's important to understand more about the functional activity of immune modulating agents that can promote the activity of the T cell subsets with the best anti-cancer ability. The phenotype and function of these lymphocytes will be highlighted, and their ability to become memory cells as well as progenitors to memory cells as they are being defined right now. And this will open the door to their specific use in immunotherapy. A second very exciting session is that on DC subsets and cancer vaccines. Probably if dendritic cells can be directly targeted in vivo, but for the time being it's also very interesting to see how ex vivo loading of dendritic cells and activation of these dendritic cells can lead to highly effective cancer vaccines. Dendritic cells continue to arouse our interest because of their central role in the initiation and regulation of cell-mediated immune responses. And this will be extensively discussed at the meeting in this session. The second subject of the session will be cancer vaccines. In general, one could say that there is ample evidence now that cancer vaccines actually work, and the specification of optimal cancer vaccines will be discussed, and in particular the advantages of synthetic vaccines compared to vaccines that are based on, for example, viral vectors. In addition, the synergy of cancer vaccines with other types of cancer treatment will be discussed, in particular the synergy with chemotherapy, irradiation, and checkpoint control monoclonal antibodies. The next exciting session is that on targeting immune suppression. In many cases, we know that the immune system is extremely tightly regulated, and this works to a great disadvantage in the case of cancer. And so we have to disrupt negative regulatory mechanisms. And in this session, in particular, regulatory T cells will be discussed, myeloid-derived suppressor cells, CTLA4, feedback control, PD-1 feedback control, IDO, which is an immunosuppressive enzyme, and nitric oxide in the tumor microenvironment will all be discussed. Then a particularly interesting session is that on immunity of oncolytic viruses. Oncolytic viruses supposedly are specifically destroying cancer cells, but nobody really knows how they work, and the possibility is that at least part of their action is based on eliciting very strong anti-tumor immune responses. Preclinical models and clinical trials with these viruses will be highlighted, and finally, oncolytic virus exploitation of immune mechanisms and the prospect of further enhancement of these responses will be discussed. A very interesting session is that on combining immunotherapy and other therapies. Current evidence indicates that already existing chemotherapy and irradiation therapy act in many cases through, at least in part, immunological mechanisms and the new knowledge gained in this area will be discussed. The same is true of certain targeted therapies that are systemic therapies that may have additional effects on the immune system, which may potentiate or counteract the anti-tumor activity of the immunotherapies. And therefore it's very important to understand this. Herewith I have just highlighted a few of our sessions, by no means all, and I hope this will all help to attract you to this very exciting meeting.